Hey everyone. So before we get started, I just wanted to say I hope everybody's okay. I know this storm was more serious than a lot of people thought it was going to be, and hopefully you and your friends and family are doing well and weren't too severely impacted by it. I know, especially in Southwest Florida, it was a very devastating hurricane. So I just hope that everybody's families are okay. So in this video, we're going to go through the immune system. I'm going to break this up into two videos. In this video, we're going to go through the innate immune system. So this will be part one. Well, part two will be the adaptive immune system, which I'll make another video for. All right, so before we get into the immune system, just to get an idea of things that could impact your immune system or cause your immune system to become activated, here's the different types of pathogens that we kind of deal with in this world. So one is bacteria. So I'm sure you're all familiar with bacteria. They're prokaryotes, they're really tiny cells. So your body usually deals with these through phagocytosis, where the bacteria is ingested by one of your immune cells and then broken down inside the cell itself. Another type of pathogen are viruses. These are particularly tricky. Viruses are not living organisms. They can't produce or replicate themselves. They have to hijack cellular machinery or your own cells in order to replicate. So we don't really find these outside the body as much as inside the cells. They're going to enter your cell, they're going to take over the cell, and then use your cell's components to either replicate its DNA or RNA, depending on the type of virus there is. So typically when it comes to viruses, we're going to need to kill the cell, kill the host cell that was infected with this virus in order to kind of kill the virus itself. Another pathogen is fungi. So we do have to deal with fungi pretty regularly. Fungi are a little tricky too because fungi are very closely related to animals. So the kingdom fungi and the kingdom Animalia are very closely related. We have a very similar common ancestor. So it's hard to find something very specific in a fungi to attack for your immune system. But there are ways, and your immune system does deal with funguses that can enter your body. Another type is protozoa. So these are usually some sort of protists. They're usually single-celled organisms, but they're eukaryotes and nice big cells. So with these, we don't really have as much phagocytosis going on as much as your immune cells are going to release some sort of chemical to break down those protozoa. And same thing with the multicellular parasites, things like roundworms or tapeworms, really big parasites in your body. Obviously, your cells can't phagocytize those. So instead, you're going to release some sort of chemicals that are going to kill the organism. So your cells release the chemicals to the external environment instead of bringing in the organism to the cell. All right, so when it comes to our immune system, we break it down into two major categories. So we have the innate immune system and the adaptive immune system. Now, the innate immune system is a non-specific immune system. It's going to involve tissues and cells in your body. They're going to try to prevent any type of pathogen inside your body or attack any type of pathogen that enters your body. So non-specific, meaning they're looking for any type of antigen that is not produced by your own cells. So non-self antigens. So that's what we're hunting for. But this immune system is pretty effective, especially with initial infestation or infection of whatever foreign pathogen there is. But then we also have the adaptive immune system, which targets a very specific antigen on some sort of pathogen that has entered your body. So this usually takes longer to initiate. When you first are infected with some pathogen that your body has never experienced before, your adaptive immune system needs to kind of prime your body in order to fight off that pathogen. But once it does, whenever that pathogen infects your body again, it's immediately ready to kill that pathogen. So the adaptive immune system takes longer than the innate immune system, but it's way more effective once it's in place. All right, so with the innate immune system, we break it down into two parts. So surface barriers are part of your innate immune system. We have lots of barriers in our body to try to keep out the pathogens from getting into our soft tissues. So things like your skin is a great example of a surface barrier, or the mucosa or mucous membrane that is around your hollow organs, all the epithelial cells. They're there to kind of create a barrier between the lumen and whatever tissues outside the barrier, again, preventing any pathogens from getting through. But once a pathogen does get through these barriers, then we have the internal defenses turn on. So these are a lot of your innate immune cells, cells that can go through phagocytosis. Also, natural killer cells are part of this category. So they're going to kill any type of foreign bacteria that's in your body. Again, non-specific, it doesn't matter what kind of antigens that are on these bacteria or viruses or anything that's entering your body. As long as it's not your antigens, they're going to try to kill it. But it's not just the cells that are involved with the internal defenses. So things like inflammation, and we also have proteins that kind of attack different types of pathogens. And even fever is a type of innate immune response. We're producing a fever to kind of kill whatever pathogens in our body. But again, all those processes are non-specific. Any type of foreign pathogen that comes in, you're going to initiate these responses, and the responses target that pathogen. All right, so focusing on the innate immune system in this video, we're just talking about what happens when bacteria or viruses or other types of pathogens first infect your body. So we call these the first and second lines of defense in your innate immune system. The first line of defense would be the barriers in your body. You're trying to keep the pathogen out, either with your skin, your epidermis of your skin, or the dermal layer or the mucosa layer lining inside your hollow organs. Again, just trying to keep out the pathogens entirely, and it's pretty effective, but not always. It's going to depend on how much bacteria is there, or virus is there, or if there's any nicks or breaches in this barrier. After that, if the bacteria or the virus or other pathogens can get into your soft tissues, that's when we have our second line of defense initiate, which is going to be those cells, the immune cells, or the proteins or other bodily functions like fever or inflammation to try to take care of that pathogen that has now made it into your soft tissue. All right, so looking at our first line of defense first, these barriers. All right, again, this is a lot of just your epithelial layers of your body, whether it be your epidermis or whether it be the mucosa in your organs. And again, they're creating a physical barrier there. Now, with your skin specifically, your epidermis of your skin, one of the other things that it can do is that it creates keratin in those cells. That keratin that your cells produce is actually really good at resisting acids and bases and other enzymes that bacteria might release to kind of try to digest and break down that barrier. All right, so it has this kind of extra adaptation to really prevent bacteria from just kind of eating their way through these cells and getting to your soft tissue where they really can proliferate and really try to take over that area. Not only that, these epithelial cells can produce a lot of different chemicals or enzymes that are going to help kill or even prevent or block some of these bacteria and viruses that are trying to get in. All right, so just to give you an idea of some things that your cells can do. One, they can release acids. Having a pretty high or low pH makes it really difficult for a lot of these bacteria and these viruses to replicate or to proliferate. So it really slows down their ability to divide, which is going to slow down their ability to infect. Also, we produce a lot of different enzymes that do similar things. So just to give you some examples of enzymes, because there's tons of them out there. Lysozymes of saliva. So in your saliva, you're producing enzymes. We have respiratory mucus enzymes that are going to be released in the mucus in our respiratory system, or even lacrimal fluids. These things lacrimal fluids have enzymes in it. All these things can kill bacteria. So when we're tearing up, one of the things we're doing is releasing enzymes in the area to prevent bacteria from infecting our eyes. Or the same thing when we're releasing lysozymes in our saliva, we're producing these enzymes that could break down the bacteria or the virus before it even gets into our system. Not only that, our stomach is used a lot for killing bacteria. So we're going to see this in a second, but your stomach is very acidic and it has a lot of enzymes in it that bacteria just don't do a good job surviving in. Same thing with viruses. I'm going to use bacteria because it's one of the major things that we have to resist, but this is true for all pathogens. Once it gets in your stomach, there's a good chance it's just going to be broken down simply from the acidity of your stomach and the enzymes that are there. So one of the things that happens in other parts of your body, like your respiratory system, it's going to produce some sort of mucus, and this mucus can catch these pathogens. So inside your respiratory system, if you remember looking at respiratory epithelial cells, one of the things that it has is goblet cells. Goblet cells release something called mucin, which once combined with water creates a mucus, which is really sticky, and that can catch these pathogens that are floating around attacking that barrier, that mucosa. And once it's collected, these bacteria in the mucus, we have those hairs that are in our respiratory system on our cells called cilia, and they're going to fan and bring that mucus upwards out of our respiratory system, where it's going to turn a corner and then go down into our digestive system, where that bacteria or that virus or rather pathogen it is going to be broken down because of the acidity of our stomach. Other things we can release too are defensins and dermacidins. So defensins are peptides that your body can release that directly inhibit bacterial growth or other microbial growth. So these are just chemicals that are released, inhibiting the growth of these microbes and inhibiting their ability to undergo mitosis. And then dermacidins is another type of protein that's released by sweat, derm in the name, right, that are actually toxic to some of these bacteria. So again, so more things that can be released that actually directly kill the bacteria, not just prevent their ability to infect. And then finally, another factor that could impact your viruses or bacteria's ability to enter your system is other microorganisms that are already living on you. So not all bacteria are bad, and they're not all out to kill you. Some of them are commensal, where they don't have any impact on your body, and your body typically doesn't try to kill these bacteria. So because we have these other bacteria growing on us, they're taking up resources that infectious bacteria could use. So since they're competing for resources, they actually help keep down some of these other bacteria that would be pathogenic and would actually cause harm in your body. All right, so going back to that whole respiratory scenario. So you might remember this from AP1, but the epithelial layer of your respiratory system is lined with that pseudostratified ciliated columnar cells. And here's a little GIF of that happening right here. All right. And you can see these little cilia that are on the surface of those cells that are fanning. And what this cilia is doing is bringing all that mucus created by goblet cells that caught microbes that are going to now fan up that mucus so it goes up and out of your respiratory system, and then it's going to turn a corner and go down into your digestive system where those microbes can be digested or broken down from the acidity and enzymes that are in your stomach. All right, so that's just another interesting case of your barriers working to kind of fend off these microbes. Now, they're not 100% effective, though, obviously, because we do get infections. So whether there's just too many microbes to use these defenses to stop the microbes, or that there's some sort of breach in this barrier, either through nuts or nicks and cuts, then you're going to have the infection come into your soft tissue, which is going to trigger the next stage of this innate immune response. All right, so the next stage of innate immune response is internal defenses. This is going to be cells that are involved and chemicals that are involved, or other enzymes or proteins that are involved. So focusing on the cells that are involved, it's a lot of the leukocytes that we've already discussed. So you know a lot of these already. But here we're going to actually introduce other leukocytes or other white blood cells that are also involved with the whole immune system. We did not talk about these before because these aren't usually found in the circulatory system. A lot of these cells are going to be found inside the tissues or connective tissues of the body, or even into the interstitial fluid of the body. So some of the ones that we have not covered yet. One is mast cells. So mast cells here. Mast cells are very similar to basophils. So if you remember what basophils do, they're really there to initiate an inflammation response. So mast cells do the same thing. The major difference between those two is that basophils are found in your bloodstream, while mast cells are found in the tissues of the body. Or specifically, they're mostly found in connective tissues. But they initially do the same thing. They release histamine, they release heparin, and other chemicals that are going to initiate an immune response. Another cell that we haven't talked about yet are dendritic cells, excuse me. Dendritic cells is another offshoot of monocytes. So if you remember when a monocyte jumps into your tissue, it becomes a macrophage where it kind of goes around looking for things to phagocytize. Dendritic cells, similar, except they're not just going to break down whatever they ingested. Instead, they're going to break down what they ingested and then take those peptides that are left over from breaking down that foreign pathogen and put those peptides on the surface of their own cells to stimulate the adaptive immune system, which we'll talk about in the next video. All right, so they're really going to be there holding up antigens to tell the adaptive immune system, hey, is this your antigen that you're looking for? And if so, come kill it. Another one we have are natural killer cells. So these are very closely related to T lymphocytes. The major differences here, T lymphocytes are part of the adaptive immune system. They're looking for a very specific antigen of a cell to kill. While natural killer cells, they're not looking for a very specific antigen. They're looking for just a general antigen, but kill in a very similar way. These are not phagocytic cells. Instead, they release different enzymes that are going to kill the cell, usually through apoptosis, which we'll talk about here in a second. Now, the reason all these are part of the innate immune system is because they have receptors that are looking for foreign antigens called toll-like receptors, but these receptors are very general. They're not looking for a very specific antigen that only one type of microbe would have. Instead, these receptors look for very common patterns that are found in almost all foreign microbes. So as long as that antigen that this is looking for does not match your own antigens, it's usually going to kill it. So there's a very broad category of antigens it's looking for, which is very different than the adaptive immune system. Again, we'll talk about in a little bit, where it's looking for one very specific antigen. All right, so looking at all these cells individually, so for neutrophils, macrophages, and dendritic cells, these are the cells that are able to undergo phagocytosis. All right, so just to give you an idea of what's happening with phagocytosis. So let's say this is one of those immune cells here. If it detects a foreign microbe, like a bacteria, and it's going to try to get rid of this bacteria, the first thing it's going to do is bring in that bacteria through phagocytosis. And simply put, phagocytosis is when you have the bacterium engulfed by the plasma membrane of the cell. And so the bacteria, or the foreign pathogen, is now stuck inside this vesicle that is created because part of the plasma membrane came with it. And so it's kind of contained inside this vesicle. So this type of vesicle we call a phagosome. It's just it's a vesicle that is holding on to one of those foreign pathogens that are now inside the cell. What happens next is that phagosome is then going to bind with lysosomes. And if you remember, a lysosome is kind of the garbage disposal of all the cells. They have enzymes in it that are there to break down different parts of the cell. Well, when the phagosome and the lysosome merge together, we call that new organelle a phagolysosome. And the enzymes from the lysosome are then going to break down the bacteria or other foreign pathogen that is inside the phagosome. So that is really where these foreign microbes are being killed, is in the phagolysosomes. Now, this whole process of breaking down the bacteria in one of these phagolysosomes is mediated by something called respiratory burst. So respiratory burst is when is a process that involves a lot of the byproducts of respiration. All right, so a lot of the byproducts of respiration, we call them reactive oxygen species or reactive oxygen containing molecules. The byproduct has this oxygen on this molecule that is very reactive to any type of other molecule. All right, so examples of this are like nitric oxide, peroxide, and superoxide. These are byproducts of respiration, and they're very toxic to cells. All right, so usually we try to get rid of these byproducts because they can attack our own cells. But with a lot of these phagocytic immune cells, they actually hold on to these toxins and release it into the phagolysosome, where two things happen. One, these reactive oxygen molecules are going to react with a lot of the chemicals that were of that bacteria and help kill that bacteria or the particles of that bacteria. And two, these reactive oxygen species usually initiate some of the enzymes that are in the phagolysosome. All right, so those enzymes are not activated until they come in contact with a lot of the products of respiratory burst. So there's a two-part function to this respiratory burst. One, it can attack the bacteria directly because they're toxic, and two, they are initiating the enzymes that help break down that bacteria. And then once that bacteria is broken down, then all the parts of that bacteria are released through exocytosis, where they go into the bloodstream, where other processes can help get rid of all those particles that were released. Now, this is true, exocytosis is true for both the neutrophils and macrophages. That's what they're doing. They're going out there, they're phagocytizing stuff, they're breaking it down, and then they're releasing the parts of that bacteria. But that's not what's happening with the dendritic cells. Instead of exocytosis happening, instead they're going to take all the parts of this bacterium, mostly the peptides that are left over from all this degradation, and they're going to hold those peptides on the surface of their cells. All right, we call this antigen presentation. That peptide is now an antigen being held up by this dendritic cell. All right, so this dendritic cell is doing this to initiate the adaptive immune system. It is going around, and different T cells and B cells are coming along and seeing if that antigen matches up with what they know, with their receptor that is very specific to a certain type of antigen. And if it does, then it's going to trigger an immune response, which is your adaptive immune system stimulating. So again, these dendritic cells are really important to initiate the adaptive immune system because of this. All right, for your basophils and mast cells, again, they're very similar cells. It's just really where they're located that is different between them. Basophils, again, are in the bloodstream, while mast cells are mostly in the tissue. They're mostly found in connective tissue, but they can be found in other tissues too. And they're both going to initiate some sort of inflammation response if they come in contact with a foreign pathogen. So a couple of chemicals that are involved here we've already talked about, like histamines. Histamines are vasodilators. Specifically, histamines really try to target the arterioles of capillary beds to bring in a lot more blood into the capillaries. By bringing in more blood, we're bringing in more white blood cells and some of the proteins that are in blood too to these areas. Histamines are also really important for increasing capillary permeability. So if you remember with some of the capillaries, if they're continuous, they don't have any fenestration, or if they're fenestrated, they have fenestrations, those pores. Well, a lot of times those are not big enough to allow some of the proteins out of the blood, or to even allow some of the white blood cells out of the blood and into the tissue. So these histamines will then increase holes or pores in this blood that are big enough for proteins and white blood cells to escape and get into the tissue. All right, so increased capillary permeability is actually a very important part of the basophil and mast cells function. We also have heparin, which is an anticoagulant. So we're going to make sure that blood is not being slowed down because of coagulation, again, increasing blood flow to the area. We also have a suite of molecules called eicosanoids, which are lipid-based molecules. So a lot of these other ones, heparin, histamine, are protein-based. Eicosanoids are more lipid-based, but they do essentially the same thing. There's a whole bunch of eicosanoids. We're still discovering more all the time. And again, pretty much the same idea. They're going to be anticoagulants or increase capillary permeability or vasodilation. And then finally, other chemicals that are released by the basophils and mast cells are called chemotactic chemicals. And these are chemicals that are going to attract other immune cells, like your neutrophils, macrophages, natural killer cells. A whole bunch of other cells are going to be attracted because of these chemicals that are released by basophils and mast cells. So they're telling your other immune cells, hey, come here, we have an infection, you need to come take care of this. All right, another type of cell is those natural killer cells that are very closely related to T cells. The major difference between those two is that natural killer cells are not specific. They're going to kill any type of antigen that doesn't match your own. Um, so we call these NK cells for natural killer cells. And again, they're natural killers because they're not looking for a very specific antigen. They're just going to kill anything that doesn't have your antigens on it. Now, these can kill a whole bunch of different types of cells. They're really important for virus, bacterial infections. They're really important for suppressing tumor cells. And this is one of the major problems with incompatible transplantation or incompatible transfusion of blood, because these cells just will kill anything it doesn't recognize. They're going to kill some of these other organs that are being transplanted into people. Now, how they're doing this is they're bouncing around from cell to cell in your body. They come in contact with all the cells in your body. And we call this immune surveillance. They're going to a cell and checking, hey, do I recognize you or not? And if they don't recognize you, they're immediately going to kill that cell. They don't care. As long as it's not your cell, they're going to kill it. But if it recognizes a cell, then it'll bounce to the next one, and bounce to the next one, and bounce to the next one. So we call that immune surveillance. They're just going around their body, surveilling different cells, bouncing into them, and seeing if the receptors can detect an antigen they don't recognize. Now, these cells don't undergo phagocytosis. Instead, they're going to release enzymes that are going to help kill that cell that is attacking. So two major enzymes that natural killer cells can release are porphyrins and granzymes, Porphyrins are going to create some sort of pore into the cell that it's attacking, the cell that it did not recognize. Now, what porphyrin does, having this now open pore, here's a little GIF just demonstrating this, but I'll show you when it pops up. Here is an example of a porphyrin, this green tube that is produced, this pore. There's two things that this can do. One, increasing the number of pores in the plasma membrane of the cell increases its osmolarity. All right, the more water that can enter the cell. And if too much osmosis occurs, that cell can burst and die. All right, so enough porphyrin can cause osmosis lysis. All right, lysis through osmosis. Another thing these porphyrins do is create a channel for other enzymes like granzymes. What granzymes do is they go into a cell and initiate apoptosis, which is programmed cell death. And this is important because instead of just having your cells explode, apoptosis breaks down the cell from the inside out and collapses the cell in on itself, where everything is broken down and held by little parts or little vesicles that are created from the plasma membrane of the cell. So here's that happening here. You can see this cell going through apoptosis. So here's the cell. These are natural killer cells injecting it with granzymes. And those granzymes destroy the cell from the inside out and cause all the parts of the cell to be encapsulated by parts of the plasma membrane, so kind of like external vesicles. Now, this is great because if this cell was infected with a virus or something, now pieces of the virus are not being flung everywhere. They are contained inside these vesicles that were created from apoptosis. And then the natural killer cells release other enzymes to tell phagocytic cells to come around and then eat those vesicles. So now everything was nice and contained and everything's being broken down appropriately. All right, and then another type of innate immune cell that we've already talked about is eosinophils. These are your parasite slayers. They like attacking those parasites that are multicellular. So since these are really big parasites, obviously we're not going to phagocytize them. Instead, we're going to release enzymes that breaks down each of the cells of this organism. And so again, I've already showed you this GIF before, but you can see all these eosinophils attacking the surface of this parasite, releasing enzymes externally that are going to then break down that organism. All right, so those are the cells involved in the innate immune response, the secondary defense mechanism. Now we're going to talk about some of the proteins that are also involved with this. So it's not just cells that are doing this. We also have chemicals that are floating around in our system that are trying to do the same or very similar things. So we call these antimicrobial proteins because they are against any microbes that come into your body. And there's two ways they can do this. They can either attack microbes directly by killing the microbe, or they can reduce the microbe's ability to reproduce. So there's tons of proteins that do this, but two major categories that we're going to focus on are interferons, and then complement proteins. All right, so just starting with the interferons, they are, again, proteins that are floating around in your blood, and they can come from different sources. So we see this a lot with viral infections. So how a virus gets into your cells, if a virus gets into your system and it's looking for a cell to infect, there's little receptors on your cells that the virus looks for. And if the virus can attach to this receptor, then it can enter your cell. All right, so that's actually important to understand, to really understand how interferons can help prevent this. So viruses are looking for receptors they can attach to, which then enter the cell, and then it infects the cell and takes over your cell to make more virus. Okay, so when this happens, your cell is a goner. It's going to die one way or another, either from the virus replicating so much that the cell explodes and flings the virus everywhere, or it's going to have the body kill itself. All right, so it's going to tell your body to say, hey, come kill me, I'm infected. All right, so either way, this is a dead cell. So what happens is that once the cell is infected, it starts producing interferons. So two interferons it produces specifically are called interferon alpha and interferon beta. And there's a few things that these interferons do. So once they're released, these interferons are produced by an infected cell. One of the things it does, it can bind to receptors of neighboring cells. And by binding to these receptors of the neighboring cells, it prevents the virus from being able to bind to those receptors, meaning the virus can't get into those cells. All right, so it kind of inhibits the virus's ability to enter the cell by binding to those receptors. Second thing that it does, once it binds to receptors of neighboring cells, it also tells those cells, hey, start making proteins that can break down viral RNA, DNA, or proteins. All right, make other proteins that can attack the parts of a virus. So even if a virus gets into this neighboring cell now this cell has defenses built into it that can help fend off that virus and prevent it from infecting that cell. And then finally, these interferons are also going to stimulate natural killer cells. All right, so it's going to tell the natural killer cells, hey, come here, I'm infected. And that natural killer cell is then going to release either perforin or granzymes. All right, and the third thing that these interferons can do is stimulate nearby natural killer cells. So they're going to stimulate natural killer cells nearby, say, hey, come here, I'm infected. And it's going to attract the natural killer cell, which will then release either those perforins or granzymes, which are going to cause apoptosis of the cell. Again, trying to contain that virus, killing the cell, the host cell, without having the virus being flowing everywhere. All right, so this is a very controlled way of getting rid of an infected cell. And then finally, another type called interferon gamma, all right, is released by the natural killer cells themselves. So these will also release a certain type of interferon. And what this interferon does is stimulate nearby phagocytes, like your neutrophils and your macrophages. And by stimulating them, they're going to come by and collect all those little vesicles that are created through apoptosis to really then digest the parts of the virus internally inside the cell, which would then be broken down into individual peptides and either released through exocytosis or, in a dendritic case, be presented on top of the cell. So again, all these cells are really working together to achieve innate immunity. Okay, so another type is called complement. There is a bunch of complement proteins, just collectively called complement. There's at least 20 proteins that we've discovered so far, but I'm sure we're going to discover more in the future because this is still pretty new discoveries that are happening as far as medicine goes. But these are a very important role in your innate immune response, more so than we ever previously realized. All right, so complements are important proteins in this whole innate process. All right, so they are activated through an enzyme cascade, and we've talked about enzyme cascades before. All right, so there are enzymes lined up, a bunch of different complement proteins lined up in a cascade that eventually stimulates a final enzyme that is going to do something as far as innate immunity goes. So the way the cascade is stimulated is through two processes. We call this the classical pathway and the alternative pathway. The classical pathway is going to require the adaptive immune system. So one of the parts of the adaptive immune system is your B cells releasing antibodies. If an antibody attaches to its known antigen, the antigen the antibody can attach to, which should be a foreign antigen in your body, then a complement cell can also attach to that antigen-antibody complex that is made. When that happens, then it's going to kind of trigger the cascade to release all the other, or to stimulate all the other complement proteins that are floating around in your plasma. But complements don't need an antibody specifically. Another thing that could attach to is the polysaccharides on a foreign pathogen. This is what's referred to as the alternative pathway. It doesn't require the adaptive immune system in order to be initiated. But again, the end result is stimulate all the complement proteins in this enzymatic cascade to eventually have some sort of response. So two pathways, classical, attaching to antigen and antibody complex, so it requires the adaptive immune system in order to do that. Alternative pathway doesn't need an antibody at all. It can attach directly to the polysaccharides that are on the pathogen. All right, so once we have these enzymatic cascades go through and we have active complement proteins going on in your body, here's a few ways that they can actually attack or help your innate immune system. One type of complement we call opsonins, which are part of opsonization. And opsonization is just a way for your complement to help your phagocytic cells in your body. So these complements, these opsonins, will attach to this foreign pathogen. And when that happens, these complements are easier for your phagocytic immune cells to recognize than just a normal antigen that would be on a foreign substance. And so what happens is it makes it easier for these cells to phagocyze that cell because it's more easily recognized than just a regular antigen of a foreign pathogen. All right, so they're just assisting phagocytosis there. Another thing they do, they're actually a part of the inflammation response. And what they're doing is stimulating those basophils and those mast cells that are nearby. All right, so if either the classical or the alternative pathway is activated, then those will automatically stimulate the nearby basophils and actophils, which causes an inflammation response. Another thing to do is cytolysis, which is again exploding a cell. All right, so this is usually done by some sort of channel being added, just like perforin. So there's other types other than perforin can also cause channels inside the plasma membrane of a foreign cell, and that increases its osmolarity. So osmosis will then fill up that cell with liquid, causing that cell to explode. And then another way complements actually help all this is eliminating cells by hitching them to your red blood cells. So if there is some sort of antibody antigen complex formed by your adaptive immune system, one of these complements can attach to that complex and then hitch that complex to a red blood cell. So now it's kind of using your red blood cells to tow this foreign body out of the area and towards areas where it can be destroyed, either by your liver or by your spleen. All right, so those are 4 different mechanisms of complements. Okay, so another part of the innate immune response is inflammation itself, all right, which is a process that involves a lot of different cells. So it's important to know the steps of inflammation to really understand what's going on here. So step 1 of inflammation is pretty straightforward. It's when your basophils and your mast cells are releasing different chemicals that are going to cause vasodilation, cause anticoagulation. It's going to cause permeability of your capillaries to increase, and it's going to attract other white blood cells through chemotactic chemicals. All right, so when that is all released through step 1, the next step is you're going to see changes in our vascular system. All right, so we're going to see vasodilation of the arterioles, where it's going to bring in more blood, all right, to these capillaries. We see increased capillary permeability, which is going to increase the amount of fluid that can be released by the capillaries into the tissue. This also stimulates things called cell adhesion molecules, which are molecules that are created by the endothelium cells of your capillaries. So here they're indicated here as triangles. And these are molecules that are going to stick inwards into your capillary, and they catch a lot of the passing white blood cells that are going by in your bloodstream. So they don't just go whizzing by. They actually have something to latch onto to stop at that inflammation area. And then collectively, all this is doing is increasing the amount of liquid and white blood cells and proteins that can get into your tissue. And we call this filtrate exudate. It's just a fancy word for the filtrate that is happening during inflammation. So we have way more filtration happening, a lot more liquid leaving the capillaries than other would be. So we call this exudate. And so we have a large amount of exudate that is bringing a lot of these immune factors into your tissues and out of your bloodstream. All right, so step 3 is recruitment of a lot of these white blood cells. So this is broken up into 3 processes. One is margination. So margination has to do with those CAMs again, those cellular adhesive molecules that are being produced by the endothelial cells of your capillaries. So you have these little CAMs that are sticking up inside your capillaries now. Well, your white blood cells also produce CAMs, and CAMs attract other CAMs. They stick to other CAMs. So what's happening is as these white blood cells are whizzing by in your bloodstream, they have these 2 different CAMs on each other that attract each other, and so it can catch that white blood cell before it just passes by. So it's almost like a Velcro for white blood cells. All right, as the white blood cell is coming by, CAM is going to attract another CAM, and so they get stuck there in that area. And again, you're only going to have these CAMs produced in areas of inflammation, areas where there was basophil and mast cell stimulation. So once that happens, these white blood cells are stuck in the area where they need to be, then diapedesis happens, which we've already talked about. Because we have higher permeabilities in capillaries, these white blood cells can now leave the capillary very easily, jump through the blood and into the tissue, diapedesis. And then once diapedesis is complete, they're going to start crawling around this tissue looking for the foreign pathogen, whether it be bacteria, viruses, or anything else. And they do this through chemotaxis, which we talked about, which is simply the white blood cell's ability to kind of hone in on areas of higher concentration of some sort of chemical. So with chemotaxis, what it's going to do is keep looking for higher concentrations. So it's going to keep following the concentration as it gets higher and higher until it gets to the source, which is that bacterium. And then it's going to ingest that bacterium through phagocytosis. So whether it is one of your neutrophils, macrophages, or dendritic cells. And then finally, step 4 is not really happening after steps 1 through 3. This is kind of happening during the same time, but it's kind of an alternate process that's going on at the same time that these white blood cells are being recruited. So we're also delivering plasma proteins in our exudate as this is all happening. And some of these plasma proteins we already talked about, like your complement proteins. So if the complement proteins come in, it's going to do a lot of the things that we say complement cell or proteins can do. All right. But on top of that, we're also bringing in other plasma proteins, ones that we already talked about involve clotting. All right, so a lot of the proteins in the enzymatic cascade of clotting come into the tissue now, and you could have clotting happen in your tissue, which is going to create clot walls that kind of block off the bacteria from going any further. Another type of protein is also being raised is kinins, and kinins are other proteins in your plasma membrane, or sorry, your plasma that can also stimulate inflammation, creating kind of a positive feedback. So as these kinins leave your plasma and get into the tissue, they're going to cause a lot of the other parts of this inflammation response to occur too, having this positive feedback, creating even more and more fluid and more exudate getting into these infected areas. All right, the last part of the innate immune system we're going to talk about is a fever. So a lot of times people think fever is kind of a side effect of being infected with some sort of pathogen, but it's actually one of your body's responses to pathogens. For a long time, we thought that it was something that we had to deal with, and that was just a symptom of the underlying infection. But really, your body is creating a fever to help fight off that pathogen. All right, so we have a fancy name for fevers in medicine. We call it pyrexia. Pyra means fire in Latin. So like pyromaniac is somebody who is crazy with fire, right? So pyrexia is producing a fire or producing increased temperatures. And we define it by an increase of 1 degree Celsius from your core body temperature, which translates to about 1.8 degrees Fahrenheit. So if you're above 1.8 degrees Fahrenheit of your body's natural temperature, then you're having a fever. And just as a reminder, our normal body temperature is about 98.6 degrees Fahrenheit. So if you go up 1.8 degrees from there, then you're having pyrexia or a fever occur. And this happens because of chemicals that are released not only by the pathogen that comes into your system that your body can detect, but also from your white blood cells too are going to release a lot of these chemicals. And we call these chemicals pyrogens. Again, pyrogens are chemicals that are going to stimulate a fever. So these pyrogens will then be sent off into your system, where they're going to go through your whole system and eventually end up at your hypothalamus, where your hypothalamus is one of the main control units of your body's internal temperature. So once the hypothalamus detects these pyrogens, it's going to do a couple of different things to increase your body's temperature. One of the things it does is it's going to constrict a lot of the blood vessels that are near the surface of your skin. By doing this, by having blood not so close to the surface, we're preventing the loss of heat from the surface of our skin. So we're kind of just tightening the core of where our temperature is going to be, which means it's not towards the surface and being lost, and the heat's not being lost to the external environment. Another thing it's going to do is increase shivering, all right? Make you feel cold, even though you're not cold on the inside, and cause your muscles to shiver. And when your muscles shiver, they're burning through ATP. And one of the byproducts of burning through ATP is releasing heat. All right, heat is a byproduct of the breakdown of ATP. And by doing this, you're increasing your body's core temperature because of all this heat being produced from the contraction of your muscles everywhere. And then finally, it's also going to increase on your metabolic activity, which again, one of the byproducts is increased heat. All right, heat is one of the byproducts for a lot of metabolic activities in your body. And by stimulating this metabolic activity, not only are we going to have more energy to fight off the bacteria, even though we don't feel like we have energy, but we're going to have more ATP to help a lot of these cells that are fighting off your the pathogen. But we're also increasing the heat that is associated with that, which is going to help with this whole process. All right, so we have 3 different stages as far as a fever goes that we define. So this is going to be your considered your onset stage, your stadium stage, and your defervescent stage. So onset is just the initial increase in temperature of your body. So again, your hypothalamus detects a lot of these pyrogens. It's going to constrict a lot of the blood vessels near your skin to help reduce the amount of heat that is lost to the external environment. You're going to have a lot more metabolic activity occur, and you're going to have shivering of your muscles contracting pretty quickly to, again, burn a lot of ATP and release a lot of heat. So your initial, again, onset is when your body's increasing in temperature. And then at a certain point, you're going to get to the stadium. Stadium is a plateau stage where you just maintain that high temperature. And that is really where your body's really fighting off a lot of this foreign pathogens that are in your body. So as you're maintaining that elevated temperature, a lot of different things are happening, helping fight off the body, but the pathogens that are in your body. But once your body gets a handle on it, then you're going to start decreasing your temperature, because increased temperature for too long can be very bad. And so that's when you start your defervescent stage if your body's starting to get a handle on that infection. All right, so some of the benefits of having a fever. One, having this increased temperature makes it very difficult for viruses and bacteria to replicate. All right, it's really hard to go through mitosis or DNA RNA replication at these increased temperatures. And it really makes it hard for a lot of the proteins that are involved with this to keep their conformation. All right, it can denature a lot of the proteins that are involved with these processes. So it's slowing down both bacterial and viral infections when this is happening. Also, this increased temperature increases the enzymatic reaction of a lot of the other enzymes in your body that are there to kill these viruses and bacteria. All right, so again, increasing the temperature increases chemical reactions, which includes the chemical reaction caused by enzymes. So this can accelerate a lot of the enzymes that we've been talking about. It can accelerate interferons. It can accelerate some of the complements. It can accelerate gamma production. Um, it can accelerate some of the parts of adaptive immune system that we were talking about in the next video. And it can even accelerate some of the enzymes that are involved with tissue repair. So you're immediately starting to repair that tissue that is there. But there are some bad parts of having a fever too. So having too high of a temperature can be a very bad thing, or having too high of a temperature for too long could be a very bad thing. So usually fevers that are over 100 degrees Fahrenheit we consider dangerous. At that point, you should start looking for help if you're having that high of a fever and it's for too long. So typically, fevers over 102 that are prolonged are going to result in a seizure. If you spike to 102, come back down, that probably won't happen. But if you have 102 and you have it for long enough, then seizures are going to ensue. So hopefully by that time, you're somewhere where a medical professional can help you with your fever. Now, if fevers get too high, if they get to 106, and it doesn't even need to be at 106 that long, you're going to start having brain damage. The tissue in your brain is very sensitive to heat changes. And if you reach 106 degrees Fahrenheit, that is when that tissue starts to degrade or the cells in your brain start to die, and those cells won't grow back. So you have permanent brain damage when you're hitting these high temperatures. And again, hopefully by this time you've already found medical help so they can help reduce your fever and make sure it doesn't get to that temperature. And then if fevers get really high, usually around 109 degrees Fahrenheit, we consider fatal. That is when your brain is going to have irreversible brain damage to the extent where it can't sustain your body anymore. And a lot of your organ systems are going to shut down at that temperature as well. So typically medical practice now is that we don't try to contain a fever anymore unless it's extreme. So we know the benefits of fever now. So we just let the person have their fever, let their body fight off these infections naturally, and then only try to combat that fever if it gets to these extreme temperatures for extreme periods of time. All right, so that is everything that we're covering in this video. So that is the innate immune system. In the next video, I'm going to go through the adaptive immune system, which is way more specific to each individual pathogen that comes into your system. It takes longer to initiate and to prime your body for it. But once your body's primed with this, it is a very powerful immune response that could really kill a lot of different pathogens. So if you're ever reinfected with a pathogen, your body can take care of it pretty quickly.